Okay, we are, whoops, let's see. Okay, hopefully this is good. Okay, we are 1 week out from our first exam. So that means those homework assignments that hopefully you've been working on, they're due at exam time next Tuesday. I will make some announcement this evening as to what exactly that exam will cover. I'm kind of tentatively thinking about it covering all of chapter 6. So we've done just a little bit of chapter 6. We're going to get more into growth and kind of environmental conditions of bacterial cells, what they like to grow in today and a little bit into next time. So I'll see where we're at today, but again, look for some announcement about that. There's one correction that I had to one of your learning objectives, just a couple of terms that we didn't talk about this semester. So I'm going to send you some announcement about that, just to put that in writing. And I will post some practice questions. So we're going to do a couple practice questions in class today that I just took from old exams. I'm going to post like 8 or 9 more, just so you kind of start to get a feel of what these multiple choice questions will look like. You should have plenty of time for the exam. I write the exams for 50-minute periods to take them in, and I'm giving you an hour and 15 minutes. So you should have a good amount of time. And I am having some extra office hours. I have a meeting at noon, but I will be in my office Friday morning if you want to come and talk about anything. Okay. So last time we started with looking at pretty intensely at the cell wall structure of bacteria, and then we kind of moved in from there, ribosomes, other structures like fimbriae and pili, and then we try to kind of compare some eukaryotic structures with the bacterial ones. Talked about endosymbiotic theory. Remember, that's that theory that tries to explain where the first eukaryotic cell came from and what role chloroplasts and mitochondria might have had there. And we just barely began chapter 12, which is kind of our survey of eukaryotes. So we're interested in microscopic eukaryotes. We just started talking about protozoa and some of the kind of characteristics that we see for most protozoa. It's a very kind of diverse taxa. But I wanted to spend a few minutes talking about one protozoa that is considered parasitic. Incidentally, if protozoa cause disease, we call them parasites. But if bacteria and viruses cause disease, we call them pathogens. So it's kind of the same thing, but we have a little different word to describe eukaryotes that cause disease than prokaryotes or viruses. Okay, so anyway, one of the pathogenic, or sorry, parasitic protozoa is this Plasmodium. Okay, and I like to spend a few minutes to talk about this. One, I just think it is so interesting. And two, I mentioned last time, this is the single greatest threat to us as Americans, at least as we travel to other countries. Malaria kills around 400,000 people a year worldwide. Over 200 million are infected with malaria. Right now, we don't have malaria in the US. There's usually only about 1,000 to 2,000 cases of people that get it when they're traveling and come back. But it has been present in the USA like in the 1800s. Why don't we get it from mosquitoes now? That's probably a complicated question, but we killed off a lot of mosquitoes when we were using DDT, like in the 50s and 60s. That kind of mosquito could certainly come back, and we could have to deal with malaria more so in the future. But it's caused, what's the cause of it? 5 different species of this Plasmodium protozoa. And don't memorize these. You should know that Plasmodium name, but the species there, you don't need to memorize. Just know that there is some difference in what malaria looks like depending on what kind of Plasmodium you've been infected with. This is probably the most famous one, Plasmodium falciparum, that causes the most severe disease. It's most prevalent in sub-Saharan Africa. And probably the second worst is the Vivex, that is mostly present in like South America, Latin America, Southeast Asia. Okay, and then the other 3 here, the malariae and the ovule, those are pretty rare. And the Plasmodium knowlesi, that's a species that until very recently we thought only infected apes. And we found a few cases of spillover where humans have actually gotten it via some kind of ape. So that's kind of rare. But we see differences in the incubation period, meaning how long do you have to start feeling sick after you bit by the mosquito, and certainly the severity of the disease. Okay, so it's a special kind of mosquito that carries these Plasmodium, this Anopheles mosquitoes. Again, they may be present to some extent in southern US. We're not seeing transmittance of malaria here right now. It's important to understand that mosquito is not harmed at all, okay, by the Plasmodium. If it would be, then we wouldn't have as much problem. We're going to look at some highlights of the life cycle in just a minute. What does the disease look like? Well, it kind of cycles through 3 different stages. And the cycle lasts like 6 to 10 hours, and then kind of will repeat itself maybe every 2 to 3 days. Okay, so it's kind of a cyclic thing. The patient, at some point, after they've been bit and there's a certain incubation time, they'll have a cold stage. So that means they're shivering. They have the chills. Okay, shaking a little bit. And then a few hours later, the hat stage, they spike a high fever, a lot of pain, like headache. They may be vomiting. Okay, and then after a few hours, basically the fever goes back to normal. So the sweating stage, so normal temp, they're sweating and they're extremely tired, fatigue. Okay, so that's kind of what malaria looks like. You might think, wow, that sounds about like the flu. That doesn't seem so bad. People have described it as feeling like they were going to die. Like the pain was so intense, the achiness, again, really high fevers. And there's a ton of different complications that patients experience. Anemia is a really common one because red blood cells are getting destroyed. Can be kidney failure, pretty common. Neurological things. I had a friend that came back from Africa with malaria, and he was in a coma for quite a time. He did wake up, and we were all kind of surprised, actually. So lots and lots of different complications there. It is treatable, especially if you catch it early. There is a lot of resistance to the drugs that we have, or at least some resistance. So I won't really get into the treatment there, but again, if you catch it early, there is treatment. If you go abroad to a place where there is malaria, your doctor's going to prescribe you some anti-malaria medication. Problem is sometimes that makes people sick to take the anti-malaria medication. So side effects. And if you don't want to go on vacation or go somewhere on a mission trip and then be sick the whole time. So sometimes it's more than people can tolerate and they stop taking it. Okay, and then they're susceptible to it. So, you know, some issues with that. We wish we had better ways to treat it. All right, so the life cycle of that plasmodium is extremely complicated. I want to point out a few things about it. You know, this is disease that's been around for probably thousands of years, and we've only kind of figured out the life cycle early 1900s. Sir Edmund Ross, I think was his name, that kind of helped figure this out. There's a lot of special names for different parts of the life cycle that we're not going to worry about. But if you start right here, this is when a mosquito that's infected with that plasmodium takes a blood meal. We always say a mosquito bite. It's not really a bite. Mosquitoes have about 5 different proboscis that they stick into you. One of them, they take a blood meal. So it's the females that take a blood meal to nourish their eggs, not the males. So they suck the blood out with one. With one of the other ones, they're injecting like anticoagulants. And I believe that's the same one that they're going to inject that plasmodium if the mosquito's infected. Okay, so a person gets bit by a mosquito, and then that stage of the plasmodium will first infect the liver. Okay, so it goes through part of its life cycle in the liver, and then kind of when the time is right, or when those, you know, like I said, we have specialized names here, don't worry about, but when that matures, it's going to enter into human red blood cells. Okay, and this is where it's doing all the damage. Here's the trophozoite stage. Remember, we defined it, we said all protozoa have a trophozoite stage. That means they're motile feeding stage. Okay, so when they're feeding, they're doing damage to us. This is where they're going to be damaging red blood cells. And this is when the patient's going to start going through that cycle of the cold stage and the hot stage, sweating. Okay, and that can kind of just go indefinitely till either the patient's immune system wins out, or, you know, they get help with some drugs to get rid of it, or the patient dies. Like any of those things can happen. As the malaria are maturing in our bodies, they actually have some sexual reproduction that they do. So some gametocytes there. And if a mosquito takes a blood meal from that patient, then another mosquito can become infected. And then we have this whole cycle that takes place within the mosquito. So there's growing, maturing, there's a cyst stage, kind of a dormant stage that can happen there. So it's complicated, but, you know, liver stage, red blood cell, and then, you know, a whole part of it in the mosquito. Okay, so that's a little bit about malaria. I did not post this, but I got this off of Bill Gates's website. If you think about the world's deadliest animal, like the first thing that came to my mind was lions, or okay, your hippos can be pretty nasty. Well, they made the list in terms of what kills people, and these are very approximate numbers, they admit. We got lions, we got hippos. Dogs actually kill a lot more people than that because they can be infected with rabies. In other parts of the world, they're just kind of roaming around. But, and snakes kill a lot of people. Humans kill a lot of people. But top of the list by far is the mosquito. So again, in the neighborhood of 700,000 people per year die of not just malaria. Malaria makes up maybe more than half of that, but there's at least 14 diseases, all of them viruses, except the plasmodium protozoa that can harm us. So things like yellow fever, dengue, chikungunya, Zika, Eastern equine encephalitis killed some people in West Michigan a few years ago. West Nile, like mosquitoes just are bad. They carry all kinds of nasty stuff. And really kind of the best we can do right now is cut down on the number of mosquitoes. So interesting. Hopefully we get better ways to treat these viral infections. I have heard it said that if we got rid of mosquitoes, like we wouldn't be any of the work, we'd be much better off. Like there's no animal that relies on mosquitoes for their life cycle. They all eat other things besides mosquitoes, that we would just be better off without them altogether. Okay, questions, comments there? Okay, if you've had malaria, come and chat with me about that sometime. Hopefully none of you have. Yes, question. Okay, so do mosquitoes do anything for the environment that makes them useful? Not as far as I know. You know, you think about animals that eat them, like bats. I think they eat other things. So that's a good question. I don't think so. Of course, it's probably something that we could argue about, though. Yeah. Right, so that's a good point. If people have sickle cell anemia, if they have one sickle cell gene and one regular allele, they have some protection against malaria. So that's, yeah, a really interesting thing. If you live in sub-Saharan Africa, you know, that would be a good thing. If you live in the US, you're better off not having any kind of sickle cell. Yeah, but that's a really interesting example. Thanks. All right, so moving on, we're just gonna take 30 seconds here basically to talk about algae. They are a kind of eukaryote that can be microscopic. They don't cause disease, so we're not going to spend much time on them. It's true that when certain kinds of algae overgrow, they can emit toxins. You've heard maybe of like red tides. But if we were in Florida, maybe we'd spend a little bit more time about that. But sometimes it happens when there's too many nutrients in the water, like the water's turned over a certain way, or some kind of sewage runoff gone into the water where it could cause that to happen. But basically, characteristics of algae, right? They're photosynthetic. They can be, it's a very diverse group. They can be unicellular. They can live kind of in colonial form, like volvox. You might have heard of that. These are diatoms. They're marine algae, or they can be multicellular, like red algae. That's what gives us agar, solidifying agent we've talked about. Or there's, huge like brown kelp, seaweed in the ocean. And it's not just that plants are big and algae are small, because this brown kelp that extends for miles and miles in the ocean. So the difference between plant and algae is kind of, you would have to know something about the life cycle of the organism. But the main idea, in case you're interested, FYI, is that for algae, every cell in the reproductive structure has the potential to become a gamete. It has the potential to kind of start a new organism, where in plants, there are parts of the reproductive structures that don't become gametes. So in case you wondered, that's the difference. We won't really go into the composition very much because it's just quite varied, but mostly they have cell walls. Again, can be silica or all sorts of different things there. Okay. Oh, another fun fact. You have just lots of interesting things to discuss over the dinner table tonight. So what is plankton, right? Like, SpongeBob doesn't really talk about this very much. But you think about it, we talk about plankton, we know it's something that lives in the water. Is it protozoa? Is it bacteria? Is it algae? What is plankton? Well, it turns out plankton are just described as microscopic creatures that live in a large body of water. All right, so they could be archaea, it could be bacteria, it could be protozoa, algae. All of those things can technically correctly be called plankton as well. So Again, just so you know. Okay. All right, so one more kind of eukaryote that we want to talk about a little bit, and that is fungi. Primary characteristics, again, they're kind of varied here. They have cell walls that are often consisting of polysaccharide chitin. Can be some other constituents there as well. So that's not always a sure thing. And we mentioned this, I think we've mentioned this already, but one other primary characteristic is that they don't photosynthesize. Okay, so that's the primary thing that distinguishes them from plants. Significance. Why do we study fungi, or what good does fungi do to us? Anybody have anything? What good are fungi? Usually the first thing that comes to college students' mind, yes? Food, yes, that's one of the first things that comes to my mind. We eat them, like mushrooms, truffles. Yeast make bread rise, right? And also drink. All right, so alcoholic fermentations are all done by Saccharomyces cerevisiae yeast. All right, what else? What else can you think of do we get from fungi? Yes, thank you, decomposition. Okay, they are essential nutrient recyclers. Okay, and if you've ever seen like a shelf fungi growing on a tree, that tree is probably not terribly healthy because it's usually living on some dead parts of the tree there. All right, so, you know, all the logs that fall in the forest, they're getting decomposed not only by bacteria, but lots by fungus as well. You think of anything else? What about if you're sick with strep throats? What about the medicine that you take? Okay, that likely has come from a fungus. All right, so drugs. Okay, a lot of antibiotics are secreted from some kind of mold. That's where we get it. Okay. Let's not forget disease. Things like athlete's foot caused by a fungus, yeast infections, thrush are some of the not too serious ones. There can be kind of lung infections, things that are much more serious. It is hard to treat fungal infections because we're trying to kill a eukaryotic cell in our body. We have less drugs that can do that without harming us. And probably one that you may not think of, some fungi are important because they form symbiotic relationships with other organisms. Okay, so an example there, I'm going to throw a term at you, see if I can spell it, mycorrhizae. Okay, these are fungi that grow with the roots of plants. Okay, so that's what this picture up here at the top is. I just cut that out of I think it was a lawn company's commercial or ad. And it's showing a bunch of grass that have those mycorrhizal fungi. So again, these are just fungi that are living among the roots of plants. That's beneficial to plants because it helps the plants absorb more water. Kind of like an extra web beyond the roots that helps the plant absorb water. So this company is saying this is good for grass. Hey, look, the grass with more mycorrhizal connections is, you know, growing more vigorously. than the grass without that mycorrhizae connection. And I don't know, those plants, those pictures don't look a whole lot different to me, but again, we know that many plants have these kind of, again, mycorrhizal kind of symbiotic relationships. So again, fungi can be beneficial. Now they can be bad, right? There can be certain kind of fungi, like the disease that killed all the elm trees in the US, that was a fungus. But they can also form these root associations that we know are very helpful and very common. Okay, so there's actually, there's two different really morphologies or shape of fungi. We've got a filamentous fungi, which encompasses molds, as well as fleshy fungi. So by fleshy fungi, I mean like mushrooms. And it's not obvious that mushrooms grow in filaments, but underground is where all the filaments are for a mushroom. I know in my yard last week, suddenly there was some mushrooms that just appeared. Well, they grow so quickly because they have this underground mass of filaments. And then when the conditions are just right, like all those, nutrients stored and water storage, and the mushrooms can pop up really quickly. Okay, so sometimes that's underground. In molds, again, they're just a bunch of filaments. The other morphology is yeast-like fungi. All right, we'll get to that in a minute. So we have a little terminology and dealing with these filaments. We call these filaments hyphae. Okay, and they're just filaments of cells. And we can describe these hyphae in several different ways. They either septate or aseptate. So A means without. So either they have septum dividing the cells, or we could call those cross walls. And this picture is a little bit hard to see, but the one on the left here, you can see these cross walls kind of here and here and here, I think. There's pores in those cross walls to allow nutrients to flow up and down the filament. Some fungi, filamentous fungi, actually don't have those cross walls. So essentially, we've got kind of one big cell here with many different nuclei. Okay, so we have a term that describes a cell that's multinucleate, and that's cenocytic. And that means they're multinucleate. All right, so hyphae are either going to have cross walls or not have cross walls, and they are also either going to be vegetative or reproductive. So we're going to call them aerial hyphae, vegetative hyphae. Okay, so a vegetative hyphae, its job is just to absorb nutrients. They're growing at the tip, and they're absorbing nutrients and water. When you get big masses of vegetative hyphae, that we have another term to describe that, we call that mycelium. Okay, so large masses are called mycelium. Okay, and so the next, I'll come back to the slide in a second. Here's an example of a fungal mycelium. It's just a huge mass of hyphae. Oftentimes this could be underground, but if it's some kind of mold, say growing on your lunch meat or whatever, then it's just there. I am almost positive, and I don't know if this was corrected in the latest edition, that this is not an SEM. because this says the units here are 3 millimeters. So that just doesn't add up. This looks like something you can see with the naked eye. Maybe a microscope would help you see it in a little more detail, but I'm pretty sure this is some kind of light microscopy. What are aerial hyphae? Like I mentioned before, these are hyphae that are reproductive. Okay, so this is going to be the source of aerial spores. Oops, I think this is an A, sorry. Okay. All right, and spores are the reproductive feature in fungi. Okay, so that's different than in bacteria. Those spores are kind of a survival mechanism. In fungi, we have these aerial hyphae that are releasing hundreds and hundreds of spores into either the air or the medium, and from those spores grow new hyphae. Okay, so that's kind of a lot of terminology to throw at you. I found this kind of nice little diagram in another source that just kind of illustrates some of these fungal structures. Spore, here's a spore right here. And when that spore germinates, you can see that the hyphae over here just germinates from that spore in a filamentous fungi. When you have a big mass of those vegetative hyphae, we call that mycelium. This term here, haustorium, I'm going to get to that one in just a minute. That's a specialized hyphae. And again, the cenocytic hyphae, there's no cross walls. It's just a hyphae that's multinucleate, and septate hyphae. Over here is an example of an aerial spore. Sorry, not a spore, aerial hyphae. And we used to go into a lot more detail about fungal reproduction. The most recent edition of your book just cut out all that information, and I was just as glad to cut that out as well. So we're not going to really look at a whole bunch of more terminology to do with reproduction, but just here's an example of a spore that can grow up from a vegetative hyphae. And when it matures and when the conditions are right, maybe a big gust of wind blows here and releases those spores. Okay, so that's one strategy. It can be either asexual reproduction or sexual reproduction. Okay, so I think this is showing these aerial spores here coming off of a vegetative spore. I'll give you another example there. This is what budding looks like. We're going to talk just kind of briefly about that. That's the reproductive strategy of yeast. Okay, so you've got kind of not an equal splitting of a cell. So like in bacteria, we get one cell, basically split it in half. But with budding, it kind of an unequal portion kind of buds off. And here, you don't worry about that. That's kind of how the hyphae grow. Okay, so that's a little bit, maybe help you with a little bit of that terminology. Not as much terminology with the yeast morphology. They're just kind of small globular cells. They're typically not perfectly round, like we would see cocci in bacterial cells. And of course, they're much bigger than bacterial cells. And they usually reproduce like budding, by budding, what I just talked about. Now, sometimes that bud doesn't totally dissociate from its mother cell. Okay, so if the bud fails to detach, then we have a special name for that, and it's called a pseudohyphae. Okay, so that's what this picture is over here. You can kind of see a bud. It almost looks like a bowling pin or something. Like the bud is starting to come off, and then it just doesn't quite detach. Why would that happen? We think that's a strategy for some yeast that may help it penetrate. Maybe into like the cracks between cells, something like that. Strategy. And know that some organisms are not either yeast-like or filamentous. It could depend on the situation, what body type the organism takes. So we call that dimorphic growth. If the fungi exhibit both yeast-like and filamentous growth. Okay, so usually it depends on the environment and what kind of morphology a dimorphic organism will take. Okay, so it might be dependent on the temperature or availability of nutrients or something like that. But in this picture, it kind of makes it look like the organism just does both all the time. You can see the round cells and the filamentous. It might be kind of switching from kind of one type to another. I don't really know. And it's not a straightforward thing like, oh, in this situation, it's always this. And in this situation, it's always that. But Candida albicans, that's a very common fungi that causes things like thrush and athlete's foot. That's just an example of an organism that exhibits dimorphic growth. Question. So is any fungus capable of dimorphic growth? I don't think so. I think it's just kind of a special thing that some can do. Yeah, so good question. Okay, so we'll just say some. Okay. Just a couple other terms that I want to talk about. If you think about, well, where do fungi get their nutrients? Right, they need to ingest nutrients in order to metabolize, in order to have energy. Most fungi are chemoheterotrophic, which we're going to pick that term apart in just a minute when we get to our chapter 6 information here. But basically that means that they are getting nutrients from organic sources. I've always found those terms to be difficult because we have like 2 different prefixes that we have to kind of pick apart there. But basically, this is what we do. We get nutrients from other living organisms. A subset of being chemoheterotrophic is saprophytic. So most fungi are saprophytic, and that just means that they absorb nutrients from dead sources. Okay, so the filaments are pretty easy to absorb nutrients from dead cells, because when cells die, their cell wall starts to break apart, and so it's not super difficult to get nutrients out of that. Okay, so again, most fungi do this, all the decomposers, but there are some that are considered parasitic, and that means they can get their nutrients from living organisms. Okay, so if they do this, they're going to use modified hyphae called Pastoria. Okay, and they didn't show you a picture of this in the book. This is the best picture I could find of this. But basically, they're specialized hyphae that can penetrate into living cells. All right, living intact cell walls or cell membranes. So they have to be a little bit more, I don't know, beefy. That's probably not a good term for it, but a little bit more strong structurally than a regular vegetative hyphae. Okay, and when fungi do cause disease, we call a fungal disease a mycosis or mycosis. Okay, so fungi can infect both plants and animals, and when they do so at the expense of that plant or animal, then we call that relationship parasitic. Okay. So again, I want to just skip all the reproductive terminology and stuff. I know that a lot of this terminology is new. Any questions, anything about fungi? We do have some upper level mycology courses here. A couple of our BMS professors they do their research with Candida albicans, so they're more interested in that sort of thing. Okay, I wanted to just throw up a couple of practice questions here before we head into some more new material. So here's a couple questions we'll take a couple of minutes with. These are questions from old exams, some fairly kind of straightforward type questions, I would say. So you're going to have a lot of straightforward questions. You're going to have a few questions that might be more application. So like knowing this, could you apply it to something else? And then sometimes questions might be like, which of the following are false? Or something they have to kind of think through a little bit more. Okay, so take a minute, think about which one of these you think is true, which bacterial morphology has an axial filament, moves the organism into a corkscrew. Just kind of straight vocabulary here. Okay, the Vibrio, sometimes the choices will be correct answers for a different question. And sometimes I might like totally make something up. So a Vibrio, that's like a more of a comma shaped morphology. Coccobacillus, where we said that one's somewhere in between a round cocci and a rod-shaped bacillus, so maybe something like that. Pleomorphic morphology has no set shape. It could be kind of anything. So the difficulty really here is telling the difference between these two. The spirillum was the more kind of rigid one that had more typically tufts of flagella at one end. And the spirochete is the one that had the axial filament kind of going down and closed into a membrane. So that's the correct answer there. Okay, and one more. Again, straightforward. What's the primary function of pili? I'll let you read that and think about what you would choose. So again, some of these answers would be correct for a different question. Like the primary function of flagella is for motility. A structure for the acquisition of nutrients, I don't know, I just made that up. Cell wall protects and gives strength to the cell. This is the correct answer. That's horizontal gene transfer conjugation. I know I haven't really talked too much about that, but that's, we think, the main function of pili. The fimbriae, the related structure, is more for attachment and to maintain a concentration gradient. That would be a cell membrane, most likely. So, okay, so few practice questions. Any other questions before we move on? Okay. All right, so we did a little snippet of chapter 6 already when we were talking about culturing bacteria in the lab. So the homework was kind of split it up into two parts, one that hopefully you've already done, and then I made the second part of it available today. Originally, I thought we weren't going to get to this, but I think we are. It's not difficult material to kind of add on here. So what are we looking at here as we go forward? What kind of requirements do microbes need to grow? So those are sorts of things that we need to understand. We're going to grow organisms in the lab for sure. Or if we're trying to kill them, what do we need to change in their environment? Environmental influences like temperature, pH, things like that. We'll explore some terminology there. Can we mathematically predict how many cells will be in a culture at a given time? Yes, we can. All right. So a little bit of numbers is the only time we really have to do that, and it will be pretty light. We can, again, if we're describing growth in a test tube, we have ways to describe that. And then how do we actually count cells if we need to do that? Okay, so if you were in lab last week, you've already had a little introduction into counting cells in the lab. So we'll just talk about some more methods there. All right, so thinking about, thought about this a little bit when we were first talking about culture medium. We think about what kind of nutrients the organisms need in order to get energy from and then to grow. So we may be doing this in a little bit out of order. Our next topic is going to be metabolism, which, you know, in the book is chapter 5. So it maybe makes sense to understand how organisms get energy. But anyway, I don't think it matters too much. So what kind of macronutrients do cells need? And macro just means they need these nutrients in large amounts. So carbon, hydrogen, oxygen, those they just need in massive amounts. And we'll just say for cell structures, because they're going to be in almost everything in the cell wall, you know, you name it. Okay, and then nitrogen, think about structures that are nitrogenous, you know, rich in nitrogen. Nitrogenous bases is one, so lots of nitrogen in nucleic acids, lots of nitrogen in amino acids. Okay, because they've got that NH2, they got an amino group there. So those are two examples, lots of things with lots of nitrogen. About 95% of a cell's dry weight is made up of those 4 elements. Okay, so that's kind of the lion's share of what is needed. Phosphorus is also considered a macronutrient because it's in things like cell membranes. Okay, phospholipid bilayers, lots of phosphates there. DNA, right? There's a phosphate that connects all the sugars in DNA. Okay, so lots of phosphorus. ATP, can't forget that. Adenosine triphosphate. And then these other elements are typically needed for enzyme function. I've got iron in parentheses there because people argue about whether to consider it a macronutrient or a micronutrient. So I just added it here, but those elements have to be associated or very commonly associated with enzymes for the enzymes to work. All right, micronutrients are no less important. Cells just need less of them. And by less, I mean that maybe tap water would have enough of that micronutrient in it for the organism. So they're also known as trace elements. Zinc, molybdenum, manganese, etc. Again, most of these are necessary for enzyme function or structural components of enzymes. And then most cells need some, at least a little bit of organic things that we call growth factors. They may need some amino acids. A lot of bacteria can manufacture at least some of their own amino acids, but they may need to get some of them in their medium. NADH, vitamin B6, again, just a few examples of some organic things that they may need. Remember, something is considered organic if it has, whoops, that's not a double bond, sorry, carbon-hydrogen bonds, then it's organic. So something, you know, I can remember at one point in my life just thinking, oh, if it has carbon in it, it's organic. Well, something like carbon dioxide is not considered organic because it doesn't have carbon-hydrogen bond. Okay. All right, so that brings us to this nifty little table that helps explain that terminology, the ways that we describe the groups that we put organisms in based on how they get their carbon and how they get their energy. All right, so if you pick this apart a little bit, the terms describing the energy source, we use two different prefixes. If an organism gets their energy from the sun, from light, we give them that photo prefix. And if they get their energy from the breaking of chemical bonds, usually some kind of redox reaction, then we give them the prefix chemo. Okay, and then we put that together with two prefixes that describe where they get their carbon source. Let me change here. If they get their carbon, auto kind of means self here, meaning they get it in the form of carbon dioxide. Remember, it's not organic, or whether they get their carbon from organic compounds. That prefix hetero means other, which means they get it from other organisms. Okay, so other meaning other organisms. All right, so again, it's inherently confusing because you've got two different prefixes. Two different categories here are quite popular. All right, we got the chemoheterotrophs. That's the category that we belong in. Okay, so we get our chemical compound, our energy source from the breaking of bonds and redox reactions like glycolysis, and we get organic compounds, carbon from organic compounds. So we're chemoheterotrophs. That's, animals, fungi, lots of bacteria. Again, very, very common. The other common category is this photoautotrophs. These are, plants and algae are getting energy from the sun, and they're getting their carbon from carbon dioxide. Okay, so those are kind of the main groups. Troph means feeding or to eat. Okay, these other two categories are mostly bacteria and archaea. They're organisms that do some really kind of bizarre types of metabolisms that, to give you examples there, I would have to go through pathways that I don't really want to take the time to do. So if you take microbial physiology, you might learn more about organisms that do some kind of strange stuff. They're environmental organisms. A lot of them live in, places like without oxygen, or, deep sea events, some kind of bizarre environments. So I'm not going to dig any deeper into those two terms. If you know that plants are photoautotrophs for the most part, and most other life are considered chemoheterotrophs, that's good enough for me. We could also add another prefix that would describe how organisms get their electrons, but we're not going to worry about that. Okay, so thinking about environmental influences that affect bacterial growth. You're going to do a whole lab on this in a couple of weeks, where you're testing different temperatures on organisms. That's usually kind of the first thing that you think of that's easy to change in a bacteria's environment is the temperature. Okay, and all bacteria are going to have some optimum temperature where they grow the fastest. And usually right above that, they start to die because, the molecules or their reactions are moving quickest at this temperature. And then soon after that, the enzymes are starting to denature and life isn't supported. But usually we have a range of maybe 20 to 40 degrees where the organism can at least survive. Okay. So of course, all right, so here's an example. I may not have posted this, but this is in a lab. They streaked out some E. coli exactly the same on temperature, you can see a little bit of growth, but not very well. 30 degrees, gosh, I don't know, that would be probably in the 80s, something like that. I forgot to look that up in terms of Fahrenheit. So better growth, and then at body temperature, it's even growing even better than that. So again, this would be more the optimal temperature for that organism. It's growing faster there. Okay, so of course we've got some terminology to describe that. I'm not too interested in the exact, degree. That sometimes is a little bit fuzzy on, the actual zone. Psychrophiles, these are our cold-loving organisms that are growing. Again, I always consider about 21, 22 being room temperature. Seems hotter than that in here right now. But 20 is about room temp. So organisms that grow, again, not just they grow, but they grow ideally. They prefer that temperature. They grow the quickest. Those are psychrophiles. You can see we've got some that are even able to grow right around freezing, which seems kind of bizarre. Mesophiles, these are organisms that they're typically growing well between room temperature and body temperature. So any organism that's causing disease in us is going to be a mesophile, most likely. So all the organisms we work with in the lab, they're all mesophiles. They're easiest to grow. Thermophiles, heat-loving organisms, somewhere between about 40 and 80 degrees Celsius as an optimal temperature. So these are not organisms you're going to encounter every day. You might have some, say, in your hot water heater at home, but these are your, hot springs organisms at Yellowstone or somewhere like that. And then we even have another category for hyperthermophiles that are growing even up, around boiling, which again seems pretty astonishing. So organisms that bacteria live pretty much anywhere. Okay, we talked about this thioglycolate medium before. We were talking about different mediums. This is a way to test the oxygen requirement of an organism. And, basically this is a medium that chemically gets rid of the oxygen. So we inoculate it, and then after we incubate it, we look to see where the organism grows. And based on where it grows, again, we label it as some, oxygen requirement. So for example, if it's only growing where there's oxygen, then that would be an obligate aerobe, an organism that has to have oxygen in order to metabolize and grow. If it does not grow at all in the presence of oxygen and only grows where there's no oxygen, it's an obligate anaerobe. Facultative means flexible here. So here's an organism with very flexible metabolism that can grow with oxygen or without. Facultatives usually will grow faster in the presence of oxygen because they get more energy when they respire aerobically. So a little heavier growth at the top, but we've got it, all throughout. And then there are these aerotolerant anaerobes that they are not poisoned by the presence of oxygen, but they don't use it either. Okay, so for example, some of the streptococcus organisms we use in lab, all they do is a particular kind of fermentation. So whether they're in oxygen or not, they don't care. They just keep doing fermentation. Okay, so they're going to grow pretty evenly whether there's oxygen or not. Okay, in practice, it's hard to tell the difference between these two categories just based on this test, but at least that's the theory behind it. Okay, and it's not actually oxygen, it's not actually O2 that poisons some organisms. Okay, so I want to talk about that in a little more depth. They do talk about this in your book as well, but O2 is actually pretty stable. There are other forms of oxygen that are much less stable that cause a lot of damage in cells, okay? Because, so if we think about why these forms of oxygen cause damage, it's because they are oxidizing agents which means they steal electrons from other molecules. Okay, so they're all strong oxidizing agents. Okay, they grab electrons from other molecules. Okay, so basically that causes a lot of cellular damage. I don't need for you to memorize the chemical formulas of these. I'll let your chemistry professors work out the chemistry of this if they want to, but you should sort of recognize at least a couple of these that we're going to talk about in a little bit more detail. So if you think about, we already talked about, well, why these forms of oxygen are dangerous. Where do they come from, and what can cells do about it to counteract that toxicity? Okay, so where do they come from? Singlet oxygen is actually formed during photosynthesis. Okay, so it's oxygen that's been boosted up to another energy level, and whatever. How do cells deal with that? They have pigments to counteract. Okay, and that's really all I'll say about that, because we don't really talk a lot about photosynthesis in this course, but that's where singlet oxygen comes from. If an organism doesn't photosynthesize, and they don't have to deal with that. Okay, the two here that we're going to kind of spend more time on here is superoxide and peroxide. Okay, both of those toxic forms of oxygen are formed as a byproduct of aerobic respiration. Okay, so I'll include that here as well. Okay, and so it's estimated that as much of about 5% of the oxygen that acts as a final electron acceptor will end up as superoxide instead of water, all right, during aerobic respiration. And we'll talk about that when we talk about electron transport chains. So any organism that respires aerobically has to have a way to get rid of superoxide. And then, as we'll see, the peroxide anion is actually related to that. Any organism that doesn't respire aerobically, they may still be exposed to superoxide just from being around like atmospheric oxygen. So I guess it's kind of everywhere, but especially when an organism's respiring aerobically. Okay, so what do cells do about that? Well, they have to have some enzyme to detoxify it. Okay, so superoxide can be detoxified by superoxide dismutase. Okay, let me erase this for a second here. Okay. All right, whoops. So this is an enzyme that detoxifies that superoxide into hydrogen peroxide and water. Let's see, almost balanced. We put a 2 right there, okay? All right, so again, that's the important enzyme. An organism has to be able to make that enzyme to detoxify superoxide. Now the problem here, hydrogen peroxide as a product, that's also dangerous to cells. That's basically peroxide anion. So cells have to have a way to detoxify that as well. And there's a couple of different enzymes that can detoxify superoxide. One of them is catalase that detoxifies hydrogen peroxide into water and oxygen. Okay, again, balance that. Okay, and this is a test that you're going to do later on if you're in lab, a catalase test. That's the production of that enzyme can help us tell the difference between certain kinds of bacteria cells. So basically you take, this is the catalase test here. You take some of your bacteria and kind of rub it on a slide dry and then you add a drop of hydrogen peroxide on there. The bubbles forming are this oxygen that's released. Okay, so if you do that test and you see bubbles, oh, then the organism must be making this enzyme. Okay, there is actually another enzyme that detoxifies hydrogen peroxide, and that's called peroxidase. And I don't memorize these reactions. But the enzyme itself is more important. Here, you don't get oxygen as a byproduct. Okay, so actually, whoops, not peroxide. I didn't leave myself very much room there. Peroxidase. Okay. So organisms have to either have catalase or peroxidase to detoxify that hydrogen peroxide. The catalase is a lot easier to test for because we've got that oxygen that we can see. I don't know how you would test for the production of water or NAD. There may be a way to do it, but okay. So the last type of toxic form of oxygen is extremely toxic, but it's usually only produced as a result of ionizing radiation. And we're going to talk more about radiation later in the course, but that's things like gamma rays or electron beams or X-rays that cause a lot of different damage in the cell. And one of the ways is by forming this toxic oxygen form. Most cells are not going to encounter that ever, if ever. So we're not really going to worry about that one either so much. All right. So the what if here is that if you look at these categories, well, an obligate anaerobe here is not going to be able to produce superoxide dismutase or catalase or peroxidase. Okay, it doesn't have the ability to make those enzymes, so they have to rely on types of metabolism that don't expose them to oxygen. An obligate aerobe is probably going to be able to produce all those enzymes. Okay, good at detoxifying toxic forms of oxygen. All right. What about pH? Obviously, that can have a big impact on growth, and we have terminology to describe that, but that's pretty straightforward. There are bacteria that occupy pretty much every niche on Earth. There are bacteria that grow optimally at low pH, so we might call those acidophiles, even down, you know, 1, that's a really acidic solution. Most organisms, are going to be neutrophiles, so they're growing optimally between a pH of like 5 and 7. And then alkalinophiles or alkylophiles, either way, they grow optimally at a little higher pH. An example, cholera, the organism that causes cholera, it actually grows optimally at a pH of 9. So a little bit more alkyl in there. There's going to be some acidophiles in your stomach. You even have bacteria that live in your stomach. pH is more like 2, something like that. So they have ways to counteract that acidity. Okay. How about physical effects of water? All right. So osmotic pressure, don't mistake this. This is not the same thing as barometric pressure. So I'm not talking about organisms that live like at the bottom of the ocean where atmospheric pressure is really high. There are organisms that can tolerate that. But what I'm talking about here is an exceedingly different solute concentration outside of the cell. And most typically, it's when there's high solutes outside of the cell. Okay, so what we're talking about is a really hypertonic medium. Because if a cell is in a really hypotonic medium, then that means water is going to be rushing into the cell. But for most bacteria cells, that's no big deal because they have this intact cell wall that's going to keep them from bursting. So if you're talking about osmotic pressure, usually we mean the pressure, or how badly water wants to leave the cell is the way that we're talking about movement there. All right, so when you're talking about high osmotic pressure, again, we mean lots of solutes in the medium. An osmophile, these are organisms that live optimally or grow optimally in areas in high osmotic pressure. So in hypertonic medium, hypertonic mediums, yeah. And it could be with like any solute. And then a halophile is a type of osmophile organisms that grow in high concentrations of salt. All right, so when I put the brackets around something, that means concentration of. Okay, so again, these are pretty extreme organisms that would grow optimally in high salt concentration. Not just like ocean water is about 3% salt, and that is hypertonic, compared to the cell. I'm talking about places like the Great Salt Lake, where salinity gets up to like 22%, or the Dead Sea, places like that. Those are when true halophiles are going to grow. There are organisms that can tolerate a little bit higher salt, for a while, maybe up to 7, 8, 9% salinity, but they're not true halophiles because they don't grow optimally. Okay, so those are organisms that have ways around losing water and have ways to counteract that. And really lastly, in thinking about kind of different environmental conditions, I'm not sure if your book brings this up in chapter 6, but I wanted to mention ecological associations because at least before the pandemic, this is a really hot area of research, trying to understand the types of organisms that live in our body is very difficult, because when we take them out in pure culture, they act differently because they're not in that mixed with hundreds or 1,000 other organisms. So to try to figure out what organisms need to be present in our body for us to be healthy or not be sick, at least, is not easy to figure out. So any kind of probiotic that's on the market, they're just guessing. Like, we don't really know yet. They're not FDA approved because we really have no idea, unfortunately, but maybe at some point we'll understand these ecological associations better. Okay, so just a little terminology to describe the relationship organisms have with other organisms. If two organisms have an antagonistic relationship, that just means one organism benefits and the other is harmed. Okay, so this is basically a parasitic relationship. Okay, so parasitic. Here's an example up here. We've got some kind of bacteria streaked on the plate, and then there's a big old mold here that you can see. There's a little zone here where now the bacteria can't grow anymore. Okay, that mold is secreting something that's keeping the bacteria from growing, probably some kind of antibiotic. Okay, so that would be an antagonistic relationship. A synergistic relationship is a little bit harder to give an example of, but that just means the members could live separately, but benefit from the association. Always let me know if you can't read my writing. Okay, so I was trying to think, this would be hard for us to know for sure about bacterial relationships, if they're synergistic. I was trying to think, well, like humans and dogs, like they can live separately, but they generally both benefit when they live together. So that's the example I came up with. Symbiotic relationships are much more common, I think, or we can maybe understand them or be sure we understand them a little bit better. That's one where if you have two organisms that are living together and both benefit, okay, then we can call that a symbiotic relationship. Here's an example of types of bacteria that live in nodules on the roots of pea plants. Okay, the bacteria fixes the nitrogen for to be in a beneficial form for the pea, and the pea gives the bacteria some nutrients. All right, so there's just some examples. We'll continue on with growth of microbes next time. Look for some communication from me about the material that will be covered next week. Okay, see you Thursday.
Studying for these.